Humans have always been explorers, across oceans, mountains, and even into space. But our most profound journey is inward, decoding the human genome, our own instruction manual. Our DNA is a vast library, a blueprint dictating everything from eye color to disease risk. Deciphering this code is like learning a new language, one that reveals our past, present, and future. The answers to who we are may be found within the intricate tapestry of our DNA. Unlocking the genome isn't just about understanding biology, it's about revolutionizing healthcare and unlocking human potential. The power of the human genome lies dormant within us, waiting to be unleashed. Our journey begins with a single cell, where DNA, the twisted double helix, resides. 4 chemical letters, A, T, C, and G, form the alphabet of our genetic code. Billions of these letters create the recipe for our unique traits. We inherit 23 chromosomes from each parent, blending their genetic legacies. Genome sequencing lets us read this code, revealing variations that influence disease. By understanding these differences, we can develop better treatments and even prevent illness. For decades, the human genome was an unreadable book. Genome sequencing changed everything, breaking DNA into fragments and decoding their order. It's like assembling a massive jigsaw puzzle, each piece crucial to the bigger picture. The genome is dynamic, constantly evolving, and even a single letter change can impact health. Sequencing allows us to spot these variations, influencing disease risk and treatment response. This technology is the key to understanding what makes each of us unique. Sequencing the genome was once slow and costly, but innovation changed the game. Automated high-throughput machines made sequencing faster and more affordable. Now, scientists can compare thousands of genomes, uncovering patterns linked to health. Projects like the 100,000 Genomes Project have provided invaluable data. This shift from single genes to entire genomes marked a new era, genomics. Genomics explores how genes, environment, and lifestyle interact to shape our health. It's a holistic approach, transforming biomedical research. Sequencing is just the start. The real challenge is making sense of the data. Bioinformatics uses computers to analyze billions of DNA letters, finding patterns invisible to the eye. These tools help identify disease-linked variations and predict individual health risks. Precision medicine tailors treatments to each person's genetic makeup. The genome is a dynamic network, not a static blueprint. Understanding these interactions is key to unlocking the full power of genomics. The Human Genome Project was a scientific moonshot, mapping all 3 billion base pairs of our DNA. Launched in 1990, it united scientists worldwide in a quest to decode our species' blueprint. Early sequencing was slow and laborious, but the potential rewards were immense. Deciphering the genome promised new insights into biology and disease. It was a bold leap into the unknown, aiming to revolutionize medicine. Driven by collaboration and determination, these pioneers changed science forever. Their work would have a profound global impact. Scientists painstakingly pieced together the genome, chromosome by chromosome. Breakthroughs in technology fueled their progress, turning a massive puzzle into reality. By 2000, a draft sequence was announced. By 2003, the complete genome was finished. The project wasn't just about data, it was about making sense of it. Powerful tools and databases democratized access for researchers everywhere. The Human Genome Project transformed how we study and share genetic information. Completing the Human Genome Project was just the beginning. Scientists use this blueprint to develop new diagnostics and treatments for genetic diseases. Genetic tests now identify risks for conditions like cancer and heart disease, enabling early intervention. Gene therapy, inspired by this knowledge, offers hope for previously untreatable diseases. The project also deepened our understanding of common diseases, revealing the interplay of genes and environment. Its impact continues to shape medicine today. Predictive genomics lets us foresee health risks before symptoms appear. By analyzing our DNA, we can spot variations that increase disease risk, like BRCA mutations in breast cancer. Early detection empowers people to take preventive action. Genome sequencing is illuminating the genetic roots of many diseases, from diabetes to Alzheimer's. Understanding these risks enables personalized prevention and treatment. Precision medicine tailors care to each individual's genetic profile. It's a shift from one-size-fits-all to truly personalized healthcare. Medicine has long struggled with unpredictable treatment responses. Pharmacogenomics uses genome sequencing to match medications to our genes. For example, genetic testing can guide safe dosing of drugs like warfarin. This approach minimizes side effects and maximizes effectiveness. Personalized medicine is expanding to cancer, heart disease, and more. Treatments are increasingly tailored to the genetic mutations driving each patient's illness. Genome sequencing is making medicine as unique as our DNA. For families grappling with rare diseases, the diagnostic journey can be a long and arduous one, often marked by uncertainty, frustration, and, honestly, a real sense of isolation. Many rare diseases have complex genetic roots, making them difficult to diagnose using traditional methods, but genome sequencing is emerging as a powerful tool for unraveling these genetic mysteries, offering hope and answers to families who have long searched for them. Consider the case of a child with a constellation of seemingly unrelated symptoms, baffling doctors and leaving parents desperate for a diagnosis. In the past, such cases might have remained unsolved, the child relegated to a lifetime of uncertainty. But with genome sequencing, doctors can now analyze the child's entire genome, searching for rare genetic mutations that might hold the key to their condition. By comparing the child's genome to those of their parents and to vast databases of genetic information, scientists can pinpoint the culprit mutation, often providing a definitive diagnosis and opening doors to potential treatments or management strategies. This ability to identify rare genetic disorders is particularly impactful for children, as an early diagnosis can be crucial for guiding their development and improving their long-term outcomes. Genome sequencing is not only transforming the diagnostic odyssey for rare diseases but also fueling research into their underlying causes and potential treatments. By studying the genomes of individuals with rare conditions, scientists can identify common genetic pathways and develop targeted therapies that address the root cause of the disease, offering hope for cures where none existed before. While genome sequencing offers profound insights into individual health, its impact extends far beyond the realm of personalized medicine. By analyzing the genomes of populations, we can gain a deeper understanding of the genetic diversity of our species, trace the evolutionary history of diseases, and develop more effective public health interventions. Imagine a world where we could track the spread of infectious diseases with unprecedented precision, identifying emerging outbreaks before they escalate into global pandemics. Genome sequencing is making this a reality. By analyzing the genomes of viruses and bacteria, scientists can track their mutations and movement across geographical regions, providing crucial information for public health officials to implement timely and effective containment strategies. Furthermore, genomic surveillance can be used to monitor the effectiveness of vaccination programs, identify drug-resistant strains of pathogens, and guide the development of new vaccines and treatments. This proactive approach to public health has the potential to save countless lives and prevent widespread outbreaks. Beyond infectious diseases, population-level genomic studies are shedding light on the complex interplay of genes and environment in chronic diseases like heart disease, cancer, and diabetes. By identifying genetic risk factors that are more prevalent in certain populations, we can develop targeted public health interventions, such as tailored screening programs or lifestyle interventions, to mitigate the burden of these diseases in high-risk communities. As we delve into the uncharted waters of genomic medicine, we face new ethical challenges that require careful navigation. Genome sequencing reveals our most personal information, raising questions about privacy, discrimination, and what it means to be human. Imagine a future where employers or insurers use genetic data to discriminate. Protecting this information is paramount. Genetic testing can also create anxiety. Comprehensive counseling is crucial to help people understand and cope with their results. Open dialogue among scientists, ethicists, policymakers, and the public will ensure genomic medicine benefits humanity while respecting our values. The human genome, that vast cosmic library within us, is finally opening up, revealing its secrets. Yet, as with any groundbreaking technology, genome sequencing faces its own set of challenges. Imagine, for a moment, a powerful telescope, capable of peering into the furthest reaches of the universe, but only a select few have access to it. That's the conundrum we face today with genome sequencing. The cost of sequencing a human genome has plummeted dramatically since the Human Genome Project, dropping from billions to just a few $1,000s. But honestly, even a few $1,000s is a significant barrier for many individuals in healthcare systems, especially in low-resource settings. It's kind of like having a life-changing book, but only a privileged few can afford to read it. This cost barrier, really limits our ability to fully realize the potential of genome sequencing for improving global health. We need to find ways to make this technology more accessible and affordable, ensuring that everyone, regardless of their socioeconomic status or geographic location, can benefit from its transformative power. Thankfully, the quest for affordable genome sequencing is well underway. Scientists and engineers are working tirelessly to develop faster, more efficient sequencing technologies that can decode our genomes at a fraction of the current cost. Just imagine a day when sequencing your genome is as routine as getting a blood test, providing invaluable insights into your health and guiding personalized healthcare decisions. That's the future we're striving for, a future where the power of the genome is within everyone's reach. Beyond the code, deciphering the language of complexity is, well, one of the greatest scientific challenges of our time. Sequencing the human genome was truly a monumental achievement, kind of like assembling a massive jigsaw puzzle with, billions of pieces. But now we face an even greater challenge: understanding what it all means. Imagine staring at a page filled with thousands of words in a foreign language. You might recognize the letters, but the meaning remains elusive. Our genome is written in the language of DNA, a complex code with its own intricate grammar and syntax. While we've made remarkable progress in deciphering the basic building blocks of this code, we're really only beginning to grasp the complex interplay of genes, regulatory elements, and environmental factors that shape our health. Think of it as a vast cosmic web, with each gene representing a node, and the interactions between them forming a complex network of interconnected pathways. A single gene rarely acts in isolation. Instead, it's part of a complex symphony of genetic and environmental influences that determine our traits and predispositions. Unraveling these complex interactions is crucial for unlocking the full potential of genome sequencing. It's not enough to simply identify genetic variations. We need to understand how these variations interact with each other, with our environment, and with our lifestyle choices to influence our health. This requires developing sophisticated computational models and analytical tools that can handle the immense complexity of the human genome. The power of genome sequencing to unlock the secrets of our DNA is both exhilarating and, honestly, a bit daunting. As we venture further into this uncharted territory, we must proceed with caution, guided by a strong ethical compass. Imagine wielding a powerful tool, capable of both great good and potential harm. That's the double helix dilemma we face with genome sequencing. One of the most pressing ethical concerns is the potential for genetic discrimination. Imagine a future where employers or insurance companies use genetic information to deny opportunities or coverage based on an individual's perceived health risks. Safeguarding genetic privacy and preventing discrimination based on genetic information is paramount. Another ethical challenge lies in the realm of genetic engineering. Technologies like CRISPR-Cas9 give us the ability to edit the human genome with unprecedented precision, raising the possibility of correcting genetic defects and even enhancing human capabilities. While these advancements hold immense promise, they also raise profound ethical questions about the limits of genetic manipulation and the potential for unintended consequences. Should we edit the genomes of future generations to eliminate genetic diseases even if it means altering the human gene pool? Where do we draw the line between therapy and enhancement? These are not questions with easy answers. They require thoughtful deliberation involving scientists, ethicists, policymakers, and the public. As we unlock the secrets of our DNA, we must remember that with great power comes great responsibility. The future of genomic medicine hinges not only on our scientific ingenuity, but also on our wisdom and compassion in navigating the ethical complexities of this powerful technology. We stand at the cusp of a new era in medicine, an era where our unique genetic blueprints illuminate the path to personalized health. Imagine a world where babies are screened for genetic diseases at birth, allowing for early interventions that prevent or mitigate future health problems. Picture a future where cancer treatments are tailored to the specific genetic mutations driving a tumor, maximizing effectiveness while minimizing side effects. This is the promise of genomic medicine, a future where healthcare is proactive, personalized, and precise. Genome sequencing, once a distant dream, is now a powerful tool in the hands of clinicians and researchers, unlocking the secrets of our DNA and revolutionizing the way we diagnose, treat, and prevent diseases. As we delve deeper into the cosmos of our genes, we're discovering a universe of complexity far beyond our initial imaginings. The human genome is not a static blueprint but rather a dynamic symphony of interacting genes, regulatory elements, and environmental influences. Unraveling these intricate connections is the next frontier in genomic research, a frontier that promises to yield even more precise and effective healthcare interventions. But amidst this technological revolution, we must proceed with caution, guided by a strong ethical compass. The power of genome sequencing to reveal our genetic predispositions raises profound questions about privacy, discrimination, and the responsible use of this knowledge. The journey of genomic discovery is not a solitary one. It's a collective endeavor, uniting scientists, clinicians, and communities worldwide. Together, we imagine a future where everyone is empowered by knowledge of their DNA, making informed health decisions. This vision of genomic medicine is more effective, equitable, and accessible to all. The human genome connects us all, past, present, and future. Our journey has just begun.